Imagine a future where children are born free from the specter of inherited diseases. Picture a world where the fear of passing on genetic disorders to the next generation is a thing of the past. Parents can rest easy knowing their children will not have to endure the same struggles they did. Imagine a world where cancer, once a terrifying diagnosis, becomes a manageable condition. No longer would a cancer diagnosis be a death sentence. Instead, it would be a treatable condition, much like the common cold. Patients would have access to personalized treatments tailored to their unique genetic makeup, increasing the chances of successful outcomes. This is the promise of CRISPR, a revolutionary gene editing tool that has the potential to transform medicine and reshape the future of humanity. With CRISPR, scientists can target and modify specific genes with unprecedented precision, opening up new possibilities for treating a wide range of diseases. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a powerful technology that allows scientists to precisely alter the genetic makeup of organisms, including humans. This means that we can potentially correct genetic mutations that cause diseases, effectively eradicating them from the human genome. This groundbreaking tool has ignited a firestorm of excitement and debate, ushering in a new era of genetic engineering. The implications are profound. Researchers are exploring the potential of CRISPR to not only treat, but also prevent diseases before they even occur. The possibilities are endless, and the excitement in the scientific community is palpable. CRISPR holds the key to curing genetic diseases, such as cystic fibrosis, sickle cell anemia, and Huntington's disease. Imagine a world where these debilitating conditions are no longer a burden on families and healthcare systems. Children can grow up healthy and free from the limitations imposed by genetic disorders, developing new treatments for debilitating illnesses, such as Alzheimer's, Parkinson's, and muscular dystrophy. These diseases, which currently have no cure, could become manageable or even curable with the advancements in gene editing technology. Patients would have renewed hope and a better quality of life, and even enhancing human capabilities. Beyond treating diseases, CRISPR opens the door to enhancing human abilities, such as increased strength, intelligence, and longevity. While this may sound like science fiction, the potential for human enhancement is a topic of serious scientific inquiry. However, this power comes with great responsibility, raising ethical questions about the limits of genetic manipulation and the potential consequences of altering the very fabric of life. The ability to edit genes brings with it the risk of unintended consequences, such as off-target effects or the creation of new genetic disorders. As we stand on the cusp of this genetic revolution, it is imperative that we proceed with caution and engage in thoughtful dialogue about the ethical implications of this transformative technology. We must consider the long-term effects on future generations and the potential for misuse of gene editing technology. The future of gene editing is in our hands, and it is our duty to shape it wisely. By fostering collaboration between scientists, ethicists, policymakers, and the public, we can ensure that the benefits of CRISPR are realized while minimizing the risks. Together, we can create a future where genetic diseases are a thing of the past, and the potential of human genetics is fully realized. CRISPR's origins lie in the humble bacterial immune system. Scientists discovered that bacteria use CRISPR as a defense mechanism against invading viruses. When a virus attacks a bacterium, the bacterium captures a piece of the viral DNA and integrates it into its own genome, creating a genetic memory of the invader. This stored viral DNA serves as a wanted poster for future attacks. If the same virus tries to infect the bacterium again, the CRISPR system uses this stored information to recognize and target the viral DNA for destruction. This remarkable defense mechanism has been honed over millions of years of evolution, providing bacteria with an adaptive immune system. Scientists quickly realized that CRISPR's ability to target and cut DNA could be harnessed as a powerful gene editing tool. By programming CRISPR to target specific genes, researchers can make precise changes to the genetic code, correcting genetic defects, inserting beneficial genes, or disabling harmful ones. Imagine a surgeon with the ability to operate at the molecular level, repairing damaged genes with exquisite precision. That is the essence of CRISPR gene editing. Using CRISPR, scientists can target specific genes within a cell's DNA and make precise changes, much like editing text in a document. The CRISPR system consists of two main components, a guide RNA and a CA's protein. The guide RNA acts as a homing device, guiding the CA's protein to the target DNA sequence. The CA's protein acts like a pair of molecular scissors, cutting the DNA at the precise location specified by the guide RNA. Once the DNA is cut, the cell's natural repair mechanisms kick in, attempting to fix the break. Scientists can exploit these repair mechanisms to introduce specific changes to the DNA sequence. For example, they can introduce a corrected copy of a gene to replace a faulty one, or disable a gene that is causing disease. Section 4, CRISPR in healthcare, a new dawn for medicine. CRISPR has the potential to revolutionize medicine, offering new hope for treating and even curing a wide range of diseases. This groundbreaking technology allows scientists to edit genes with unprecedented precision, opening up possibilities that were once the realm of science fiction. From genetic disorders to cancer, CRISPR is poised to transform healthcare as we know it. Imagine a world where genetic diseases can be corrected before they even manifest, or where cancer can be targeted at the molecular level, reducing the need for invasive treatments. One of the most promising applications of CRISPR is in the treatment of genetic diseases. These are conditions caused by mutations in our DNA, and until now, many have been considered incurable. Scientists are currently exploring the use of CRISPR to correct genetic defects that cause diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. By directly targeting the faulty genes, CRISPR offers a way to fix the problem at its source. By correcting these genetic errors, CRISPR could potentially cure these diseases at their root. This means that future generations could be free from the burden of inherited genetic disorders. CRISPR is also being investigated as a potential treatment for cancer. Traditional cancer treatments like chemotherapy and radiation can be harsh and have significant side effects. Scientists are developing CRISPR-based therapies that can target and destroy cancer cells while leaving healthy cells unharmed. This precision could lead to treatments that are not only more effective but also less debilitating for patients. These therapies hold the promise of more effective and less toxic cancer treatments. Imagine a future where cancer can be treated with minimal side effects, allowing patients to maintain a better quality of life during and after treatment. Beyond treating diseases, CRISPR could also be used to enhance human health and longevity. This could include everything from boosting our immune systems to slowing down the aging process. Scientists are exploring the use of CRISPR to boost the immune system, making it more effective at fighting off infections and diseases. This could lead to a future where our bodies are better equipped to handle health challenges, slow down the aging process, potentially extending our healthy years and improving our quality of life as we age. This could revolutionize how we think about aging and longevity, and even enhance human capabilities. From improving physical and cognitive abilities to potentially unlocking new levels of human performance, the possibilities are endless. As we stand on the brink of this new era in medicine, the future looks incredibly promising. Section 5. CRISPR in Agriculture. Cultivating a Sustainable Future. CRISPR is not just transforming medicine, it is also poised to revolutionize agriculture, offering solutions to some of the world's most pressing food security challenges. This groundbreaking technology allows scientists to make precise changes to the DNA of plants, enabling the development of crops that can better withstand the myriad challenges posed by our changing world. From increasing crop yields to developing disease-resistant plants, CRISPR is ushering in a new era of sustainable agriculture. By enhancing the genetic makeup of crops, we can ensure that they grow more efficiently and produce higher yields, which is crucial for feeding a growing global population. One of the most promising applications of CRISPR in agriculture is the development of crops that are resistant to pests and diseases. Traditional methods of pest control often rely on chemical pesticides, which can have harmful effects on the environment and human health. With CRISPR, we can create plants that have built-in resistance to these threats, reducing the need for chemical interventions. By editing the genes that make plants susceptible to these threats, scientists can create crops that are naturally more resilient, reducing the need for pesticides and herbicides. This not only helps in protecting the environment, but also reduces the cost of farming, making it more accessible and sustainable for farmers around the world. CRISPR can also be used to improve the nutritional content of crops, making them more nutritious and beneficial to human health. Imagine a world where staple crops like rice and wheat are fortified with essential vitamins and minerals, helping to combat malnutrition and improve public health on a global scale. For example, scientists have used CRISPR to develop tomatoes with increased levels of lycopene, an antioxidant linked to a reduced risk of heart disease and cancer. These advancements not only make our food healthier, but also open up new possibilities for combating chronic diseases through diet. Furthermore, CRISPR can be used to develop crops that are more adaptable to changing environmental conditions, such as drought, salinity, and extreme temperatures. As climate change continues to alter weather patterns and create more extreme conditions, the ability to grow crops that can thrive in these environments becomes increasingly important. This is particularly important in the face of climate change, which is already impacting crop yields around the world. By harnessing the power of CRISPR, we can develop crops that are not only more resilient but also more productive, ensuring food security for future generations. The potential of CRISPR in agriculture is vast, and as we continue to explore and refine this technology, we move closer to a more sustainable and secure future for all.